Welcome back everyone. I am Saptaparni from BioLive. Hope all of you are doing very well. So in today's video we are going to discuss about genetic cross in Drosophila, and today we are going to focus on vestigial wing mutation in Drosophila. So in my previous video I have talked about eye color mutations in Drosophila and how you can derive the parental genotype from the given F1 progeny phenotypes. Okay, so now in today's video we are going to focus on wing mutation and specifically vestigial mutation. Okay, so first of all we must remember that the vestigial mutation is an autosomal recessive mutation. Okay, the vestigial gene, the gene responsible for vestigial mutation, is on second chromosome of Drosophila. So it is an autosomal gene, okay, or an autosomal mutation, and it is recessive in nature. It is recessive in nature means when both the second chromosome contains VG mutation, then only the Drosophila will show vestigial wing pattern or vestigial wing or atrophied wing, okay, reduced wing. Okay, and with that we must also remember that we denote vestigial mutation as small vg and normal or wild type wing as vg plus, okay, small vg plus. Okay, now let's come to questions. The first question asks, see, these are the F1 progenies, okay, and these are their phenotypes and numbers. There are 10 vestigial male, 11 vestigial female, 29 wild type male and 30 wild type female. Now you have to derive the P generation cross from the given F1 progeny data. Okay. Now, as I have already told you in my previous lesson that whenever you get this type of question, the first thing you must do is to draw their genotype. Okay, so now let's take a look. It is vestigial male. Okay, now it is a male, so first of all we should draw the sex chromosome. It is the X chromosome with all wild type genes or alleles, and it is the Y chromosome as it is a male. Now it is vestigial in nature. Its wing is vestigial. That means both the second chromosome or both the autosomes of this fly must contain mutant allele, that is VG allele, vestigial allele. Okay, as we know that vestigial is a recessive mutation. So to express the recessive phenotype, both the autosome must contain the mutant allele. Okay. Now second thing is vestigial female. Now as it is a female, obviously there will be two X chromosomes. So these are the two X chromosomes with wild type of alleles. Okay. And as it is vestigial, so obviously both the second chromosome or both the second autosome of these should contain VG mutation. Okay. Now wild type male. First of all it is a male, so obviously there will be a X chromosome and Y chromosome, and X and Y both are wild type in nature. Now it is wild type. Okay. Now that means either both the second chromosome can contain VG plus, that is the normal or wild type allele. Okay. Or one autosome may contain VG plus and another autosome may contain mutant allele VG. Okay, and obviously there will be X chromosome and Y chromosome here too. Okay, so there could be two types of genotype for wild male. Now wild female. Now as it is a wild female, so obviously X chromosome, both the X chromosome will be wild type, and as it is a wild type, that means both the second autosome can contain either VG plus, that means the wild type allele, or one of the second autosome may contain VG, that is the mutant allele, and another one may contain VG plus. Okay, in heterozygous condition obviously it will show wild type phenotype. As we know, the vestigial is a recessive type of mutation. Okay, so in heterozygous it will also show wild type phenotype, and obviously there will be 2 X chromosomes, wild type X chromosomes here. So these two can be the wild type females genotype here. Okay, now first thing first, first of all we must derive the sex ratio from here. So male is to female equal to, how many males are there? 10 plus 29, that means 39 males, and how many females are there? 11 plus 30, that is 41. So it is 1:1 in ratio. Okay, now I have told you in my earlier lesson that whenever the sex ratio is 1:1, that means there is no lethality in the F1 progeny. Okay, that means the cross will be of one type when there is no kind of lethality, and when there is lethality, then the cross will be of another type. So there is no lethality, that means all the F1 progenies are alive. Okay, now let's take a look on how their phenotypic ratio differs. So here wild type wing or normal wing versus vestigial or reduced wing equal to, how many of them have wild type wing? See here, 29 plus 30, that means 59, and here vestigial wings 10 plus 11, that is 21. Okay, so 59:21, that is 3:1, which is the monohybrid cross ratio. Okay, now let's do the cross here, derive the parental generation cross here. So whenever it is a monohybrid cross, okay, that means there should be, both the parents should be of heterozygous type. Okay, whenever it is a monohybrid cross, just remember that the parent should be of heterozygous type. Okay, now why? Let's see. See, there are vestigial male and females. Okay, and in case of either vestigial male or female, one vestigial allele is derived from father and another one is derived from mother. Got it? So obviously one of the second chromosome of mother must contain VG allele, and obviously one of the second chromosome of father will also contain VG allele. Okay, and obviously for mother there will be 2 X chromosome as sex chromosome, and for father one X chromosome and one Y chromosome for as sex chromosome. Okay, now the other allele or the other autosome of mother or the other autosome of father must be VG plus here. If it is not VG plus, then these phenotypes cannot be achieved. Okay, these phenotypes, these 4 types of phenotypes in 3:1 ratio. See here the ratio would be 1:1:3 cannot be achieved. Okay, so the cross must be of here in the cross the female, the father and mother should be of heterozygous type. So now let's derive the gametes from here. So obviously there will be 2 types of allele from mother. This is the mother's, the allele should be VG and plus and VG plus plus. Okay, now from father there would be 4 types of allele. One is VG plus, see here VG plus, another one should be VG with Y chromosome, VG with Y chromosome, then VG plus with wild type of X chromosome, VG plus with wild type of X chromosome, and then VG plus with Y chromosome, VG plus with Y chromosome. Okay, now if we draw a Punnett square and if we put all the male gametes on the top side and female gametes on the left side, then we will get 8 progenies in F1 generation. Okay, what will be their phenotype then? Here see VG plus plus, that means as it is VG, both the autosomes contain VG, that means it is a vestigial fly. Okay, and as there are 2 X chromosomes, so it is a female, so it is a vestigial female. Similarly here also you can see both the autosome contains VG mutant type of allele, so it is a vestigial fly, and as it has 1 X chromosome and 1 Y chromosome, so it is a vestigial male. Then VG plus, that means in heterozygous conditions, so it will be wild type or normal winged fly, and 2 X chromosomes, so it is a wild type female. Then VG plus, that means it is wild type and X chromosome Y chromosome, so it is a wild type male. Then VG plus, it is wild type and 2 X chromosome, so it is a wild type male. Vg plus Vg, that means it is wild type with X and Y chromosome, wild type male. Vg plus Vg plus, that means it is wild type obviously. In homozygous wild type alleles are present here and both X chromosome, so it is a wild type female. Then Vg plus, both the autosome contains wild type of allele for vestigial mutation, so it will be obviously wild type and as it contains X and Y chromosome, it will be wild type male. Okay, so you can see here there is one vestigial female, one vestigial male. Okay, so one vestigial male and one vestigial female, whereas there is three wild type of female and three wild type female and three wild type of male. Okay, three wild type of male. So you can see here, comparing with the question, the ratio 1:1:3:3 is now achieved. See here, the ratio 1:1:3:3 is now achieved. So our cross is correct here. Okay, got it? So our cross is completely correct. Now coming to the second question. See here, the F1 progenies have 20 vestigial male, 20 vestigial female, 21 vestigial male, 21 normal or wild type female and 20 normal female, 20 normal male. Okay, so you have to derive the P generation cross. So first thing first, we have to derive the genotypes. As it is a vestigial female, so obviously both the autosomes will contain mutant Vg allele, and as it is a female, there will be 2 X chromosome. So next, vestigial male, so obviously both the autosome will contain vestigial mutant allele, and as it is a male, so there will be a X and Y chromosome. Now normal female may contain, in normal female, both the autosomes may contain either wild type of allele, that is Vg plus with 2 X chromosomes, or one autosome may carry Vg plus allele, the wild type allele, and another one may carry Vg allele, that is the mutant type of allele. And as we know that Vg plus is dominant over Vg, so obviously in heterozygous condition only Vg plus's phenotype will be shown. That means it will be of wild type. Okay, as we know that Vg is recessive to Vg plus. Okay, and obviously with that 2X chromosome should be there. And in case of normal male, the thing is same. Either it could be 2 autosomes with Vg plus allele and X and Y chromosome, or it could be one autosome with Vg plus allele, another with Vg allele, that means heterozygous wild type, this one with X and Y alleles. Okay. Now first thing first, we have to derive the sex ratio, and here you can see male is to female ratio would be, how many males are there? 21 plus 20, that means 41. And how many how many males are there? 21 plus 20, 41. And how many females are there? 20 plus 21, 41. So the ratio is 1:1, that means there is no lethality, so all the progenies of the F1 generation is alive. Now let's find out the phenotypic ratio, that means normal is to vestigial winged fly ratio. How many vestigial flies are there? 20 plus 21, 41. And how many normal flies are there? 21 plus 20, 41. So it is also 1:1, 41:1, 1:1. So it follows the test cross ratio. Okay, so here easily you can derive the P generation cross. Okay, as we have known that it is a test cross ratio, so it is very easy to determine the P generation cross. See, first thing first, obviously the vestigial female or male, whatever, they contain 2 Vg alleles. That means one Vg allele is from father and one is from mother. Okay, so obviously both the father and mother will contain one Vg allele, at least one Vg allele in their autosome. Okay, so see here in female mother, we can see one Vg allele on one chromosome, and in father we can also see one Vg allele on one autosome. Okay, and obviously for mother these are the 2 X chromosome, and for father these are the X and Y chromosome. Okay, now what would be the allele for the second the another autosome for both father and mother? Okay, so So from the phenotypic ratio we can see that it is following the test cross ratio. So there should be there should be one individual from father or mother who will have a homozygous recessive condition in their second chromosome or in their autosome. So because we know that in test cross we cross the homozygous recessive one with the F1 generation. Okay, so obviously there should be a homozygous recessive one individual with homozygous recessive genotype. Okay, so let's take the female or mother as homozygous recessive with both the autosome having VG or mutant type of alleles. Okay, and the father one is heterozygous VG with VG plus. We can also take the reciprocal cross, that means where the female will be of VG with VG plus. Okay, one chromosome with VG and another with VG plus, and whereas the dad or the father both autosomes should be of VG. Okay, we can also take this cross. You can also do this cross by yourself and you can see the result is same here. Okay, so from mother, obviously now I am crossing this is not a part of this cross. Now from the female we can get only one type of gamete, that is VG with plus. Okay, and from father we can get four type of gametes: VG with X chromosome, see VG with X chromosome, VG with Y chromosome, VG with Y chromosome, then VG plus with X chromosome, VG plus with X chromosome, and VG plus with Y chromosome. So VG plus with Y chromosome. And when we will do the Punnett square, we can see that we have drawn the male gametes on the top side and the female gamete on the left side, and we can see four progenies are formed, and the first one has VG and plus, that means it is a vestigial female. Then it has VG with X and Y, that means it is vestigial male. Then next is VG plus and VG, that means it is wild type, as we know that VG plus is dominant over VG. So in heterozygous condition only the phenotype of VG plus will be shown, so it is a wild type or normal winged Drosophila, and as it has two X chromosomes, so it is female. And then VG plus VG, it is also wild type, but it has X and Y chromosomes, so it is a wild type male. Okay, so here you can see the ratio is 1:1:1:1:1, which matches with the given ratio in your question, that is also 1:1:1:1. So obviously our cross is correct here. Okay, our P generation cross is correct here. Okay, so I think you have got this crosses and there is no confusion in that. In my next video we will discuss about the Carli mutation, which is also a wing mutation in Drosophila, and the related crosses. Okay, you can also watch the video of eye color mutation. I will give the link below in the description box, and I will also provide it at the end of the video. You can also check out there. Okay, so if you find this video useful, don't forget to hit the like button and share it with your friends, and don't forget to subscribe to my channel for more such videos. Thanks for watching and happy learning.